Okay, so good morning, everyone. Thank you all for being here. Also, first, thank you to the jury members to agree to evaluate my thesis and also being on the jury, and also all the in-person members who came here despite the rain, and also all the remote attendees who were able to make it. So today we are here I'm here to present my thesis work titled Towards Automatic um, Spinal cord MRI analysis for improved estimation of imaging biomarkers. Yeah, so this is the outline of my talk today. Because the talk is going to be focused on spinal cord imaging, I'll give a brief introduction to the spinal cord anatomy, and also introduce medical image segmentation in general. We'll also look at what are some of the MRI biomarkers that we want to measure from spinal cord pathologies, and why do we need segmentation tools. Then we'll move on to the contributions of the thesis, followed by the summary, the clinical impact, and the acknowledgments. Okay, so the spinal cord is part of the central nervous system consisting of both the brain and the spinal cord, and it acts as a communication pathway between the brain and the rest of the body. It is divided into 4 regions: cervical, thoracic, lumbar, and sacral, and there are some nerves that are coming out from each of the regions that direct different bodily functions. So this is a cross section of the spinal cord. We can see that there's a butterfly-shaped structure, which is the gray matter, surrounded by the white matter, and then the nerves that we saw before are coming out of this cross section. But from an image analysis perspective, we are interested in measuring the morphometry of the spinal cord, which is essentially like measuring the size or shape or area, volume, etc. And these give us insight into some of how the quantitative measures are changing according to different demographical factors such as age and sex and so on. And some of the morphometric measures that we'll be looking at are shown as example below. Okay, so this is an example of how an MRI of the spinal cord looks. So these are 2 contrasts, T1 weighted and T2 weighted. And the structure of interest here is the long tubular structure, which is the spinal cord, and which is surrounded by the cerebrospinal fluid. And again, a cross section of the same, and this is the circular thing, which is the spinal cord, and we are interested in segmenting that. Okay, so before moving on to the image analysis of the spinal cord, let us look at what medical image segmentation means. So it is essentially partitioning raw image data into meaningfully structured regions. So this is an example that is showing the segmentation of different organs in the body across different imaging modalities, CT, ultrasound, MRI, etc. And it's useful in monitoring disease progression, or getting some risk assessments, or even computing some image-based biomarkers. And the promise of automatic image segmentation is that it saves time by assisting radiologists in prognosis and possible diagnoses as well. So the most common architecture used in image segmentation is the UNet, as we can see in the right. And several variants have emerged over the years, modifying different parts of this architecture. For example, changing the backbone with a ResNet network, or even adding some attention modules in the bottleneck, which is this part, or even replacing the entire encoder part with the transformer encoder and so on. But in the space of medical image segmentation, and in UNet deserves a special mention. This is a self-configuring segmentation framework that employs some rule-based parameters to abstract away some of the architectural design choices that we usually have, which has a number of layers that we need, patch sizes, and also kernel sizes of the convolutions, etc. We have seen over the years that this framework is extremely robust and continues to achieve state-of-the-art results over novel architectures on several different data sets. Okay, Now, let us look at what are some of the biomarkers in MRI in spinal cord pathologies. So this is an example of uh, MRI in spinal cord injury. And this study says that neuroimaging biomarkers are predictive of clinical outcome in SCI patients. And this study also says that because of the lack of automated methods, uh, these measures are usually done manually, and that is time consuming as well. And another example of a chronic disease is multiple sclerosis, where spinal cord MRI can help, and it does have diagnostic and prognostic value. Specifically, if we look at spinal cord atrophy, which is the loss of the spinal cord substance, it is used as a tool to monitor the progression of MS as a disease. And so there's clinical value in the detection of subtle atrophy in MS patients. So the question now is, how do we measure these biomarkers? We need segmentation tools that can mitigate inter-rater variability that is arising from manual annotations, in the sense that if you have multiple raters doing the segmentation, there's bound to be some variability, and we would like to mitigate that with automatic methods. In most cases, the studies that do measure these quantitative biomarkers are typically small scale, because of the bottleneck associated with manual annotation, and having an automatic tool would help scale these studies to large multi-site cohorts. Okay, so given the background and the motivation for this work, so the overarching theme of this thesis was to look at how can we develop robust and generalizable segmentation tools that can better estimate imaging biomarkers in spinal cord MRIs. And a related sub-theme is how can we ensure that the segmentation accuracy achieved by these models actually correlate with clinical utility. So looking at the first contribution, so this SCIseg is a tool to automatically segment lesions in spinal cord injury on T2-weighted MRI scans. This work was published in Radiology Artificial Intelligence Journal. So spinal cord injury is an acute, it refers to an acute damage to the spinal cord, which is typically resulting from some trauma, meaning like a fall or a sports-related injury or a car accident, etc. And so the traumatic event dislocates the vertebra, and then that usually results in the compression of the spine, as we can see, compression of the cord, as we can see, and also in some cases, it also tears the spinal cord. And this is an example of the MRI of the spinal cord. And you can see some dislocation of the vertebrae and also a lesion forming on the spinal cord. So as a side note, SCI is usually a devastating event for the family and the patients. It has adverse socioeconomic consequences, because the patients are usually become paraplegic or tetraplegic depending on where the injury occurs and how high the injury occurs on the spinal cord. So, and as I said before, MRI biomarkers in SCI are useful in predicting the status of the patient's outcome. And because of the lack of open source methods and studies trained on single site data that do not generalize well, there's a need to segment SCI lesions. But it does not come without its challenges. Typically, the images are noisy, and it is very hard to properly locate the lesion on the spinal cord, and it is also there's also quite a lot of noise from metal artifacts that are implanted to support the spine after the injury. And to add to that problem, there's the appearance of the lesion also evolves over time. So this is an example of how the lesion has progressed from slightly bright to more bright. In clinical terms, of be getting more hyperintense as we go from the day of injury. So that is an example of chronic lesion. And as I said before, because we need to stabilize the card once the injury happens, there are some metal implants that are inserted, and that typically interfere with the segmentation, as algorithms usually focus on segmenting the implants and so on, which is not good. So given this, the objective of this work was to develop a model to automatically segment the spinal cord and the lesions in traumatic SCI. To go about this, we gathered a large data set containing about 190 patients, and they span the injury level is across the spinal cord, so cervical, thoracic, and lumbar, and it does contain both good resolution and bad resolution images and all different types of lesions as well. For the training, we used a three-stage approach. In the first stage, from a small unlabeled, a small labeled sample of the data, we trained an intermediate model. Based on the unlabeled set that we have, we got initial segmentations from this intermediate model and then refined them using the human in the loop active learning approach. And then we added them back to the training set to do a final multi-site training to get the final network, which segments the spinal cord and the lesion, as we can see at the bottom here. Now, I briefly also mentioned that we are also interested in ensuring that these tools are clinically useful. So we went to the literature and looked at some of the MRI-derived quantitative metrics. These are some examples: intramedullary lesion length, lesion volume, and also the axial damage ratio. So we computed these measures from the segmentations that we obtained and validated our model with that. So these are some qualitative results. We can see that the model, because it was trained on this data, it segments the lesions and it segments the spinal cord in the presence of lesions compared to the previous methods. And also the similar case for lesion segmentation, where the 3D model was able to obtain better segmentations than the 2D one. So these are qualitative results, but what is interesting from this work was that the biomarkers that were automatically derived, they significantly correlated with the clinical scores. So yeah, so for example, in this, we are plotting the axial damage ratio that I've just mentioned. And on the x-axis, we have a pinprick score. So pinprick score is a score that measures patient's neurological status after the injury. So higher the pinprick score, better the sensory function of the patient. So ideally, higher, that means that they are recovering better. And with this result, what we wanted to show was that once we compute this axial damage ratio on manual segmentations and automatic segmentations, are they correlating well? And this is what we see in this plot. So in yellow, we have the measurements obtained from manual ground truth. And in green, we have the measurements obtained from SCIseg, our model. And we see that they're generally correlating well with what has been done manually. Similar is the case with another quantitative biomarker, in this case, the lesion length, and so on. So this is one of the interesting results from this work. So the takeaways overall is that SCIseg is the first tool that that segments the spinal cord and the lesion in SCI. Open source tool, I mean, sorry. It was trained on a diverse data set containing multiple different image resolutions, sagittal, axial, different lesion types and injury phases that I showed before. And we found no significant difference between the lesion metrics derived manually and automatically, which is good, which means that the segmentations obtained from SCIseg are reliable. And this work is accessible is integrated into Spinal Cord Toolbox and the model can be accessed on any scan using these commands to get the segmentation and also to compute the morphometrical measures that I showed. Okay, so the previous work was focused on one pathology, spinal cord injury, and one contrast, T2 weighted. But In this work, we look at the spinal cord segmentation at different, across different MRI contrasts. And why are we doing that? I'll explain that. So this work was published in Medical Image Analysis, and it is titled Towards Contrast Agnostic Soft Segmentation of the Spinal Cord. Okay, so there are multiple sequences in spinal cord MRI. This is an example of the several different sequences, and we can see that the spinal cord is appearing darker and brighter in some contrasts. And all these sequences have their own purpose. For example, in some cases, the lesions are appearing better than the other contrasts, and they can be used for further analysis as well. And also the resolution of these images is also changing. So at the bottom, you can see some images are pixelated. So they are low resolution images. So the issue with having multiple sequences is that when you compute the cross-sectional area of the spinal cord, it varies depending on the MRI contrast. So we are interested in this measure. So once we get the segmentation, we compute the cross-sectional area. The problem now is, for an example, if you look at two contrasts, T1 weighted and T2 weighted, we pass them to a segmentation model, we see that the area computed from a T2 weighted model is approximately 8% higher than that computed from T1 weighted. And this is a problem. But why are we interested in CSA in the first place? So CSA is an important biomarker in several spinal cord pathologies, particularly to characterize spinal cord compression, in this case, degenerative cervical myelopathy. And it is used to also compute spinal cord atrophy, which I mentioned before, especially in MS, where detection of spinal cord atrophy is critical because it helps in better stratifying patients, or even reducing the size of clinical trials used to compute the spinal cord, used to detect atrophy in patients. So overall, it is a biomarker which is used in several spinal cord diseases. But why does it vary across MRI contrasts? What is the problem? It turns out that segmentation algorithms are typically sensitive to partial volume effects. So this is an example of partial volume effect in the spinal cord. And it refers to the phenomenon where you have multiple tissues, the intensities of multiple tissues getting averaged within one voxel, which is not representative of the true intensity of the underlying anatomy. In this case, if we look at this red box, which is 1 pixel, the value of this pixel turns out to be the average of the blue tissue and also the CSF, and that is resulting in this average intensity. And segmentation algorithms have a hard time in properly segmenting the spinal cord because of the partial volume effect. And another limitation of the existing methods is that they produce binary segmentation masks, which do not account for partial volume. So for example, so by binary masks, I mean it's either a 0 or 1 value, and we can see this abrupt transition between the cord and the CSF when we have binary masks. And that is one of the reasons why we have different CSA for different MRI contrasts. So the objective of this work was to develop a model that reduces the variability in spinal cord cross-section area across different MRI contrasts. To go about this, we used a public database of about 260 participants, and each participant had 6 contrasts, and these images were obtained from different MRI vendors. And the key contribution of this work was a novel pre-processing strategy that went from hard contrast-specific masks to a soft segmentation mask that has float values between 0 and 1 for each contrast. So the pre-processing strategy contains 5 steps. The first step is to obtain a spinal cord segmentation using the existing model. these are hard ground truth, and we manually correct them on contrasts. And then we register all of these contrasts to a common space, in this case, a T2 weighted image space. And once we register the image, we take the warping fields that we obtained and bring the segmentations also to this common image space and average them to obtain a soft mask. And we bring the contrasts back to the native space by using inverse warping fields. So we went from, as I said before, we went from contrast specific binary masks to a unique soft mask per participant per contrast. Okay. For training, we used a soft seg approach. So before that, so this is the typical pipeline for training a segmentation model. You have a ground truth, you apply some data augmentation, you binarize them again, using one hot encoding. You pass them through the network, you apply some activation, and then you use pixel-wise classification laws such as dice losses to optimize your network. What SoftSeg did was to do away with this binarization and let the model train on soft segmentation masks. So basically, implicitly obtaining soft masks while training and replacing the pixel-wise classification laws with the regression laws, which is the adaptive wing. So the idea of having a regression-based loss function is that the model is trying to learn to output float values, basically. So if a pixel is has a smaller intensity, it learns to output a smaller value between 0 and 1 and not 1 directly. So we adapted the SoftSeg approach because in SoftSeg the ground truth was created only from a single mask. We replaced that with the soft masks that we have obtained with our pre-processing, and we kept the same pipeline to train the model and then output a soft mask. Okay, so the training network is a simple unit. No bells and whistles here, just multiple input contrasts, and then the output is a soft segmentation. So for the validation, we used this intra-subject CSA as the metric. What this is essentially once we have the segmentation, we take the slices in the C2, C3 vertebral level, and we average the area of the cord across these vertebral levels, and we plot them. And what we see here is what we obtain, the cross-sectional area obtained by training on hard masks. So the first step in the pre-processing. So you can see the violent plots are not on a straight line. There's a variability across contrasts, and this is what we wanted to reduce. While training with soft masks, we can see that the discrepancy between contrasts has reduced. And this is what we achieved by, so this is what I mean by training on soft masks reduces CSA variability. We also looked at the most commonly used T1 and T2 weighted contrasts because these are the most commonly used ones, and how these are, how it is affecting, how the soft training is affecting the result. So if we look at the most commonly used method, which is DeepSeg, we saw that it is, as I said before, it is overestimating the CSA. But with the soft approach, we can see that it is bringing closer to the diagonal line. So yeah, the diagonal line is basically having, the closer to the diagonal line, the better the model, because that means that it is the same CSA for T1 and T2. Yeah, recall that we trained only on healthy participants, and also this contrast did not exist in the training set. But once we tested it on different, even on pathological patients and also different contrasts, we saw that our approach still obtained good segmentations compared to the previous approaches that we compared with. And similar is the case with another pathology as well. So what we found in this work was that training on unique soft masks reduces CSA variability. Soft masks, as I explained before, they better account for partial volume effects compared to the standard binary ground truth. This work is also accessible, is open source and accessible in Spinal Cord Toolbox. Okay, so moving on to the third work. This is titled Monitoring Morphometric Drift in Lifelong Learning Segmentation of the Spinal Cord. This is currently under review at the Imaging Neuroscience Journal. Okay, so we observed that medical data sets are not static. They typically grow over time as more data are acquired, and models are trained with different versions of a growing data set. To make this concrete, this is the evolution of how the spinal cord segmentation models developed in this toolbox. So we started with SCT PropSeg, which was trained on healthy controls, and then we moved to DeepSeg SC, which was trained on healthy controls in MS patients on 4 contrasts, and our previous work, contrast agnostic, which was trained on 6 contrasts. So we have increased the number of contrasts, and the models are also evolving. But at the same time, there are several specialized models in the literature. For example, one for MS, one for spinal cord injury, and one for DCM. These are either trained on single contrast. They have different strategies for training. So essentially, the models for spinal cord segmentation are not standardized. So in this work, some of the questions that we asked is, can we create one model that can segment most contrasts or pathologies? Instead of having multiple different models, how can we update this model when we have new data? And how can we monitor or quantify performance drifts between different versions of the segmentation model? So these questions collectively take a broader view of lifelong learning in some sense, where we do not focus on learning new tasks, but we want to focus on the life cycle of a single model that is deployed to address a real-world task, in this case, the spinal cord segmentation. So the first objective was to create a model to segment a wide range of MRI contrasts and pathologies. So we curated a large data set of about 1,600 participants, 9 MRI contrasts spanning across 6 pathologies and different vendors and image trends of the scanner. And these data were curated from 75 clinical sites, so it is pretty diverse in that sense. And for the annotation, we use the semi-automatic approach, which involves manual correction, also quality control. So for the pathologies where we saw that model already existed, a pathology-specific model already existed, we used that to get the segmentations. And for unlabeled data sets that we obtained, we used the semi-automatic, like the human in the loop active learning approach, where we got the initial segmentations, refined them, and then added back to a training set. And once we have that, we have a final model that segments the cord that was trained on all 1,600 participants. Okay, so but in this work, we are also focusing on what happens after creating the model, right? We want to have a pipeline that could automatically monitor performance drift across model versions. So we took inspiration from what we see typically in software development, where we build a model, we test it, deploy it, have some tests to check for failures, and also monitor its performance over time. So in a similar vein, we trained the model, deployed it as a release on GitHub, and we used the intra-subject CSA, which I explained before, as the metric we want to track these models across. And we have a workflow which is triggered once we upload the model. So for what this does is that it downloads the data which is hosted on Gitanix. It runs inference on the test set and computes the cross-sectional area, and also automatically plots the drift between the current and the previous versions. So this is a big overview of what I explained. So we start from unlabeled data, have a automatic and manual approach to label them. We aggregate them into a large data set, train the model. Once it is deployed, the monitoring is done, is taken care of automatically by the GitHub Actions workflow. So this is how the interface looks. This is the workflow that I explained, downloading, running inference, and plotting the results. And because we are using public resources, we do not have access to GPUs in the free version. So we have to make sure that the inference is also running fast enough that can be computed across batches. And once this is done, all the results are cached, which are then pulled to plot the plots. And this is the interface of how a release looks. So we have the model here and the code that is associated with the model. And once the workflow is done, the CSA of this model, v3.0, is automatically appended to the release along with the plots. So what this means is that, let's say if we have v4.0, v5.0, they automatically fetch this CSA, the CSV file, and also use that to plot how the model is evolving across time. As a metric, we are now using the standard deviation across the CSA. So why standard deviation? Because we have 6 contrasts that we want to measure. And then lower the CSA, the better the model is at in segmenting different contrasts similarly. So we have seen that our current model, which is v3.0, is not significantly different compared to what the previous model achieved, which is good in this case. And note that this was trained on a much larger data set. So this model is also able to segment a diverse range of contrasts and pathologies while obtaining similar performance as the previous state of the art. Okay. So with this work, we now have a tool to segment the spinal cord in a much better way. We have used software development practices for automatically monitoring morphometric drift. Similar works have been done, but on a different premise, meaning that they wanted to bring they wanted to build machine learning models with a view of doing open source software. So basically version tracking the model weights or parameters and so on. We took a different approach where we kind of track the model after it has been trained to monitor the drift between different versions. Okay. So, and there are some limitations of this work. So it is currently not fully automatic in the sense that the criteria for retraining is judged qualitatively. So we need to look at the segmentations that the model obtained on difficult cases to understand whether it is good enough for the current version, or we need to go back to train it again. Unlike the previous version, it does not output soft masks. This is a trade-off that we had to make because it is difficult to obtain soft masks in cases where we don't have multiple contrasts per participant. And it also assumes that there's a public test set that is available to be downloaded. In our case, the public database only contained healthy participants, so this was okay. But future works could also have pathological data in the test set to be able to compare the drift more accurately. Okay. So what have we achieved over the course of these works? So these works collectively present a step towards robust and generalizable tools for spinal cord and lesion segmentation. All the models that we have trained were trained on a diverse data, including multiple resolutions, noisy and good images and so on. We have repeatedly used the human in the loop active learning approach to alleviate bottlenecks that are associated with data annotation, making use of existing models and also adding our models in the loop. We have achieved state-of-the-art performance using simple, rigorously validated CNN architectures, which echoes the findings of a growing list of works which say that a well-tuned CNN still outperforms or performs similarly compared to, say, transformers or novel state space models at a fraction of the training cost. And this is an important point. We made sure that we go beyond generic segmentation metrics because segmentation metrics average out the the performance on difficult cases, which are essentially which are useful in judging whether the model, the tools can be translated to clinical practice. So there is a need for clinically targeted measures and to validate the model on clinically targeted measures. In all our works, we identified a biomarker which could benefit from segmentation, and we validated our models against that. Finally, segmentation models are more than snapshots in time. We designed a framework that could automatically monitor drift across models. Okay, so we have found that the collaborators and clinicians who shared the data with us have found the tools useful. These are some testimonials of some good feedback that we obtained from the models we developed. And it was also interesting to see external validation of the segmentation tools. For example, this study used SCIseg to measure mid-sagittal tissue bridges. So what these are, so once we have the segmentation of the lesion, we can compute the width of the tissue that is adjacent to the lesion. And this is also an indicator of recovery in SCI patients. And this can be automatically computed now with SCIseg. And what they found that the segmentations from the model are reliable compared to what was obtained with manual segmentations. This work also used SCIseg, but on a different cohort of DCM patients to compute lesion morphometrics. Okay. So it has been 5 years since I started my research, so I'm on my way to climb the academic ladder. I've been fortunate to obtain several scholarships and travel grants to present my work, and also a research grant to visit Technical University of Munich in 2024. And I've been invited to give 2 talks, and also my work was featured in 2 newsletter interviews. Yeah. And yeah, I mean, as they say, PhD is a journey, and it has been one. I'm grateful to my friends and colleagues whom I have met from all over the world. And there was a lot of good times, a lot of sport events. And I mean, I made sure that I would like to have an active sport life during a PhD, and that happened because of shared interests between me and my colleagues and also supervisor. So yeah, so that was good. And also in the end, I would also like to thank my co-supervisors and supervisor like Julian and Surath for the time that they've invested in my progress. And I've learned a lot from them during the 4 years of the PhD, and also the advice that they have given me over these years, both career-wise and research-wise. And I am grateful for that. And thank you to all my friends at different labs, and also all the funding sources of RQNT, Unique, Quebec Bioimaging Network, and DARD for funding this work. Yeah, thank you.